Welcome, everybody. Thank you very much for joining us this evening. My name is Charlotte, and I'm the Public Engagement Manager here at ZSL. And I'm joined this evening by Dr. Katharina Seilern-Moy, who is a wildlife vet here at ZSL, and she's going to be talking to you more about her work with the Garden Wildlife Health Project, and in particular, how monitoring garden wildlife health can be helped with citizen science, so sort of the involvement of citizen science in that.
Thank you, Charlotte. Also, thank you to Ali and Lyndsey for organizing this, and thank you all for listening tonight. I'm going to try and share my screen now. I hope this is visible to everyone now. Please let me know if it isn't or if there are any other issues. So yeah, thanks again for listening tonight and for organizing this great event. Great to have so many people here tonight. As Charlotte mentioned already, I'm going to briefly talk to you tonight about citizen science and how we use this to monitor the health of garden wildlife, so animals that we find in our garden. So my name is Katharina. I'm a wildlife vet for this project and also a research associate. And I've been with this great organization, ZSL, since 2016. All right, let's get started. Sorry, there we go. Okay, so why do we study wildlife health and diseases? What's the purpose and what do we actually do with all this information? So there are in general three main objectives. One of them is to monitor what's already there, so any endemic diseases, those that are known to be present in the country and might potentially already be affecting our wildlife. The second one is to detect new diseases or diseases that we haven't seen for a while or weren't aware of their presence. And the last one is to be prepared. What do I mean by that? I mean to perform horizon scanning. So keep an eye on the horizon for diseases that haven't yet arrived in the UK, but might have the potential to have an impact on our wildlife if they were to come into the country. And all of this we do with the view to conserve biodiversity and protect wild animal welfare, but also to safeguard our public health and any companion animal and livestock health, and to document potential wildlife crime and help prevent persecution. So what is citizen science actually? So if you type in citizen science into Google, you will get all sorts of definitions in all colors and forms. But in summary, citizen science is scientific work, for example, data collection, that is undertaken by the members of the general public, so whoever wants to participate. And this is often done in collaboration with or under direct supervision of professional scientists or scientific institutions. So wildlife disease surveillance schemes have the overall goal to detect a range of infectious or also non-infectious disease threats, and they're typically reporter-led, and therefore they rely on soliciting reports of observations of sick or dead wildlife for their investigations. And citizen science offers a very powerful tool to achieve this form of disease surveillance, and it's particularly well suited for any charismatic wildlife species or species that are frequently observed and positively perceived by the general public. So there are many advantages to citizen science, and some of them are listed here. For example, if you participate in citizen science, you will hopefully gain a certain level of scientific education. Participation encourages you to engage with nature and therefore promotes the general well-being. And you participate in public health improvements as well as in optimizing habitat management for the well-being of wildlife, as well as for yourself or ourselves. There are, of course, also huge benefits to science. So it allows us to collect large-scale data over long periods of time across huge spatial distance. Especially when we think about gardens, we as scientists would never be able to monitor what's going on in everybody's garden without the help of the general public, as we obviously can't be there ourselves. So Garden Wildlife Health is the project that I'm working for, and that's an example for a citizen science scheme. And the fundamental aim of our project is to monitor the health of and identify any potential disease threats to British garden wildlife. But there are, of course, many different aspects to this. For example, once we identify disease threats to British wildlife, be it in a single species or also in a whole taxonomic group, we try to be also actively involved in finding and promoting ways of mitigating certain threats wherever appropriate. And this can vary in the scale, of course, from providing individual members of the public with advice on what they can do in their own gardens to reduce a certain disease threat, up to developing national mitigation strategies to deal with, for example, any invasive species or new pathogens that might have come into the country. So the species that we are focusing on in general is garden birds, including birds of prey, as well as amphibians, reptiles, and the hedgehog is the only mammal that is involved in our project. So whilst the project is led by ZSL, where we do all our disease investigations, we work in close partnership with the British Trust for Ornithology, the BTO, with Froglife, and with the RSPB, the Royal Society for the Protection of Birds. Why do we do that? Mostly to draw from their knowledge on the natural history of all the species that we're performing surveillance on, but also to benefit from their huge supporter and membership bases and their authoritative voices in terms of all the public outreach that they already do. And particularly with regards to the BTO, they have a proven track record of citizen science that's incredible, and which is basically the foundation of the Garden Wildlife Health Project. But of course, in order to learn as much as possible and to reach as many people as possible, we collaborate widely with many other organizations from a range of sectors, including governmental organizations, conservation NGOs, wildlife gardening organizations, rehab centers, academic institutions, and many other organizations wherever possible. Okay, so how does Garden Wildlife Health work now? So here's a brief schematic introduction of what we do, and I will go into detail in the following slides. So in general, with the help of the public, we undertake scanning surveillance for infectious and non-infectious diseases that might be affecting wildlife in British gardens. And to facilitate the study of the diseases we might identify, we build sample archives, and we collaborate with a wide range of organizations, as I just mentioned, and then we integrate our findings with population monitoring in order to determine any potential impact the disease might have on a population level of a certain species. And ideally, this leads to us being able to provide people with evidence-based advice on disease mitigation, or also habitat management or other best practice strategies for their own gardens. So there are, of course, many different methods of conducting wildlife disease surveillance. We within the GWH project in general, as I mentioned, we rely on the public to report disease incidents or also incidents of fatalities of garden wildlife to us, and people do that via our website. And occasionally, if there is a freshly dead carcass involved, we can also obtain a specimen for postmortem examination. So we receive these disease reports in two different ways. So one of them is the opportunistic reports, and the other one is the so-called systematic reports. So what does that mean? So briefly, I'm going to tell you about the opportunistic reports and how that works. So the opportunistic reports are so-called ad hoc reports of members of the public reporting sightings of sickness or death to us via our website, which is shown here in a screenshot where you can see the reporting button. And this leads you to the next page where you can create your own profile, which takes a few minutes, and you can log the report as and when necessary whenever you observe them. There's a quick form to fill in where you can add details on the species that you have observed, the number of animals that might be affected, any signs of illness you might have observed. And there's also a section to add additional notes or queries, and which is also really helpful. There's the option to upload photos if you were lucky enough to take one. And during this process, you will also be available to tick a certain box if there is a freshly dead carcass available, which will prompt one of us veterinarians to contact the reporter regarding the possibility of the submission of the carcass for examination, which will all be with all the costs being covered by the project itself, also the submission. So the opportunistic or ad hoc report, where members of the public can report sightings to us as they come, means that we gain a maximum potential observer coverage. And this also offers the best potential way to detect any novel incidences or conditions that might occur. But as so many things in life, this also has a few issues or disadvantages. For example, there is a potential bias associated with observer effort, meaning that you will, for example, likely receive more reports from areas with a denser population or where more gardens are present. And there's also a potential bias towards certain conditions that might have just received a lot of press coverage, as people will, of course, suddenly be more aware of what the condition might look like, that it's out there in the country, and they will even actively try and spot this specific condition. Like on this newspaper article, you can see great tits affected by avian pox virus, which leads to this typical tumor-like growth on the head or body of the bird, and which is, of course, very easily spotted once it reaches a certain size. There often also is a species bias, meaning that more charismatic species, like the robin or the well-beloved hedgehog, are far more likely to get reported than potentially less popular species. Other types of bias could include incidences of unknown causes, as people are less likely to report a case where they are kind of certain what the cause of death is. So, for example, cat predation or acute trauma like window collision of a bird. However, we need to consider that there remains the possibility of an underlying infectious or non-infectious condition that might have predisposed the animal to such a traumatic event in the first place. So it's actually also really useful to us to receive reports of these obvious causes of death. So opportunistic reporting occasionally also means that people report after the event has actually happened, which means that we can't obtain a carcass should there have been one involved, and we might lose some valuable data from these reports. And importantly, with opportunistic reports always concerning individual or isolated cases, there remains a certain uncertainty around the impact that any suspected or detected condition might have on the species on a population level. So overall, we can conclude that whilst opportunistic reporting is extremely valuable and provides us with lots of hugely important information, there remains a certain lack of statistic validity to the data that we can collect, and therefore we are limited in some of the conclusions that we can draw from this. So we need a different system to add in. Because of these limitations, we also use a network of systematic reporters, and these are consisting of over 15,000 garden birdwatch participants, which are citizen scientists also, which is spread across the whole country, and this garden birdwatch scheme is organized by the British Trust of Ornithology. So the garden birdwatch participants, they report incredibly on a weekly basis regarding the presence or absence of different species in the gardens, and that contributes towards an incredible abundance data. And whilst they're reporting these species, they can also include information on any sightings of sickness or death, or they can also indicate if there are no signs of ill health observed, which is a very valuable piece of information, because it's very different from I haven't looked to I actually haven't spotted anything that looked unwell. So all this provides us with some statistical validity, and our disease surveillance combined with this population data that provides us insights into potential impacts that a certain condition might have on the population of the species. I also wanted to briefly mention that as a response to the national lockdown that we're all going through at the moment, and people spending more time in their gardens, the BTO Garden Birdwatch, who are also celebrating their 25th anniversary this year, they made a participation in this Garden Birdwatch scheme free of charge for a 1-year membership. And I thought I'll share this with you in case you're interested to join or know anyone who might be interested to join. So it's a good way to try it out now that it's for free. Okay, so whether opportunistically or through the systematic report, members of the public, as I mentioned, report to us via our website. And that allows us to collate all the necessary data that we require in order to conduct the disease surveillance that we need. So wherever possible, we try to assign a so-called syndromic incident definition, meaning a suspect diagnosis, which is based on a set definition that we have developed over years, but which of course we never can confirm without actually having performed an examination on the affected animal. These suspect diagnoses often relate to diseases that are easily identifiable by the clinical signs they might cause or by the species involved, like we can see here on the left. And as I mentioned before, avian poxvirus infection in tit species especially. This causes this very typical growth on the head or body of the bird and are very easily spotted and identified. Or here to the right, there is a chaffinch that's affected with the very species typical chaffinch leg lesions, which are caused either by a papillomavirus infection or a parasitic mite infestation, or possibly even a combination of both. The reporters also have the option to submit a photo, which can often be very, very useful because, as you can see here, something that is sort of seen as a growth on the head of the body might actually on a photo then turn out to be a tick, which very rarely happens, but you can see how valuable these photos can be. Assigning syndromic conditions, or these so-called suspected diagnoses, as we call them, is of course not always straightforward. So the more information we receive with these reports, the more likely we can categorize and understand what might be going on. For example, here, what you can see on the left is a fluffed up bird, a fluffed up blue tit, that doesn't tell us anything other than a bird feeling unwell or potentially in winter just being cold. However, if you look at this case, a bird being fluffed up with food stuck to the beak, showing signs of difficulty swallowing while still trying to feed, and then being of a relevant species, like the greenfinch here, that would be an indication for a bird being affected by FINCH Trichomonasis, to which I will get into more detail later on. Okay, so whilst these suspected disease definitions give us a lot of information in order to be certain of what the cause of disease or death actually was, as I mentioned before, we need to perform a postmortem examination. So if the report involves a dead animal, and should the carcass be considered to be in a suitable condition for such an examination, there is the option to offer this carcass during the reporting for examination, in which case we will contact the reporter or submitter directly and explain the submission process in detail, provide them with a submission label, and also discuss any potential public health precautions that should be taken. And as I mentioned before, the submission and the examination, the costs for that are all carried by the project. So what do we actually do during these examinations? So we conduct a full gross examination of the carcass and all the organs and body systems in our labs at the Institute of Zoology at London Zoo. We also perform additional diagnostics, which almost always include microbiology and parasitology. And then depending on the species or the findings, we can conduct further tests like molecular diagnostics or histopathology, or even sometimes radiography if needed, which often might be useful in birds of prey. The overall aim of all of this is to determine the cause of death, of course. However, we also try to identify any potential underlying or other conditions that might have been affected the animal, but might not have been directly contributing to its death. Additionally, and that's also very important, we want to gather general data on morphometrics and find out what is actually normal in this different species, like what pathogens can be found in a certain species without causing disease, as many of the species we deal with are actually still quite understudied. So these two maps give you an idea of the coverage that we have achieved using this systematic and opportunistic reporting system. So these are combined data. And as you can see, we have received reports of sick and dead garden wildlife from more or less the whole of Great Britain. And the gaps you see likely reflect the bias whereby, as mentioned before, you're less likely to receive reports from areas where human and wildlife habitats don't really overlap. Very similar distribution. So on the left, we see the disease incident reports, and on the right, the reports where we actually received carcasses for examination, and there is a very similar distribution. And also to mention, the vast majority of the reports that we receive involve garden birds, and that is then followed by amphibians and hedgehogs, and which is often seasonal, and we only receive very few reports on reptiles. I think around 1% maximum. And this is likely to the very secretive nature and them just being really good at hiding. However, it's really important to know that the information we gain from these reports and examinations on reptiles is extremely valuable for us to learn more about the conditions that might be affecting them. It's really valuable. Okay, so we use the reports and postmortem examination findings to identify and to especially study the infectious and non-infectious diseases that are affecting the animals. And the postmortem examinations enable us to conduct extensive sampling of tissues from the specimens we examine. And over the last 25 years, we have created one of the largest wildlife tissue archives in Europe. And these archives are really, really great resource to facilitate our research. Like, for example, if we were to detect a certain disease or pathogen for the first time, this archives enables us to look back through previous cases to see if this specific disease might have been around before already and was just overlooked. And if it was, then for how long was it there for? And this helps us to identify the emergence events of diseases and to basically trace them, trace their spreads through time and space. So when working especially with citizen science and with the general public, we think that it is really essential that this is a two-way street. So when we receive reports or any specimen for examination, we always communicate back to the reporters and we provide them with information on what we think might be going on or might be causing the observed signs, or what the cause of death is after we've examined the animal, and also inform them about any potential related public health information or concerns, any potential advice about disease mitigation, habitat management, wherever applicable. So one way of providing such information and the evidence-based advice is through our disease fact sheets. We have a disease fact sheet library on our website available free to look at and also free to download as PDFs, which you can see here is the upper part of the lungworm fact sheet in hedgehogs. And these fact sheets contain information on the pathogen that might be causing a certain disease, the species it might affect, the signs it might cause, as well as any public health concerns or advice on how, if there were a disease outbreak, one could mitigate and stop the spread of the disease. We also, of course, try to publish any new findings in scientific journals. And wherever possible, we aim to publish those in open access journals to make our research available to the wider public. And they're all accessible on our website as well, as you can see on the right side here of the screenshot of our website. And we also have an interactive map on our website, which allows people to look into the spatial distribution or also the number of certain disease conditions that we have observed, or look just at the species that are mostly reported to us. Here is a quick example. Here are mapped all disease incident reports and postmortem examinations of hedgehogs, comparing 2 consecutive years. So on the left, we see 2018, and on the right, 2019, whereas the circles indicate the disease reports and the triangles where we actually got a specimen for examination. So in addition to trying to advise and communicate with the general public, we also provide information and advice to inform NGO and government decision-making. And one example, or one of the best examples for this, is our participation in the Great Britain Wildlife Disease Surveillance Partnership. So that's a partnership that convenes twice a year to discuss any current or potential future disease threats to British wildlife and whether any potential action could be taken. And between all the organizations that participate within this partnership, we cover the vast majority of all native species, and it's a great way of avoiding any overlap or duplication of effort. We're also trying to be as active as possible on Twitter and Facebook. Social media is a very important tool when you want to reach many people. We try to share any recent findings or seasonal advice, but also any news from partner organizations, collaborators, or any other related information. And as you can see on the right side here, we also provide our reporters with a biannual e-newsletter that you will receive via email once you're reported to us. And that offers any up-to-date summaries of our research findings, public outreach events that we've participated in or are planning to attend, and any other relevant project updates or useful information for the time of year. When possible, and also in order just to be more visible to different groups and different audiences, we try to get involved in various outreach events to communicate and interact with a variety of people. And we also try to collaborate on articles and press releases with Nature or Wildlife magazines or any other organizations. It's just important to stay visible and communicate. Okay, now I wanted to quickly give you 2 examples of how we use garden wildlife health projects and what we've learned. So one example will be focused on a specific disease in garden birds that was detected through this project and how we've learned about its effects. And the second example will be on how we gain an understanding of a certain conservation issue. In this case, I'm going to focus briefly on hedgehog declines. So FINCH Trichomonasis, as I briefly mentioned before. FINCH Trichomonasis is a parasitic disease caused by a protozoal, meaning a unicellular microscopically small parasite called Trichomonas gallinae. And the disease is predominantly affecting greenfinches and to a little lesser extent also chaffinches, but has been reported in most finch species and also many other garden birds. This infectious disease causes severe lesions in the gullet of the affected bird and leading to an obstruction of this gullet, basically preventing the affected bird from being able to swallow. The birds will then, of course, still attempt to feed, but will now can't swallow, so they will drop the seed again, which is covered in saliva containing the parasite and offering therefore an easy way for the next bird to get infected when it just comes along and picks the same seed up that the infected bird just dropped. So affected birds, due to the nature of the disease, and these birds are mostly greenfinches and chaffinches, they will typically appear fluffed up and lethargic, which are general signs of a bird just being unwell. And then they tend to be observed trying but failing to feed, and then often with a messy face and food and saliva stuck to the beak or their faces. So this disease was first identified in British finches in 2006, and it raised a lot of public concern, given the large number of sudden death in greenfinches and to a lesser extent, as mentioned, chaffinches, as seen in this picture. And it's probably one of the best examples of an emerging infectious disease, meaning a disease that wasn't previously present in a specific species. And this disease had a dramatic impact on the greenfinches in Great Britain, as I will show you here. So it is so far suggested that since its incursion into the greenfinch population in 2006, this finch trichomonasis has caused about a species decline of about 70% in the breeding population of British greenfinches, which is just an incredible impact on a population. However, although the disease has only relatively recently emerged in British finches, it is important to note that the pathogen, or the parasite in this case, is not actually new. So it has been well documented and known for hundreds of years as a cause of ill health and also a cause of death in columbiforms, meaning pigeons and doves. as well as in birds of prey that might feed on those. So whilst we aren't absolutely sure about this, the whole story seems to fit with the theory that in the human adapted landscapes that we're living in, and in this case particularly the popular practice of garden bird feeding, which of course has lots of positive effects on bird populations, species are brought together that would normally have very little interaction, and that might have provided the parasite with an opportunity to jump species and therefore affect a new host, in this case with a devastating effect. And now once the disease is established in the new host, the parasites can just continue to circulate within this species because they basically, of course, feed together. So in case garden birds are affected by this disease, or also mostly other diseases in our gardens, whilst we can't treat animals in the wild, there are measures we can take to help reduce further disease transmission in garden habitats. So we therefore always recommend, where multiple birds seem to be affected by certain disease, and especially FINCH Trichomonasis, to temporarily stop feeding during disease outbreaks, to stop birds from congregating and sort of encourage them to disperse. Of course, to always follow good feeding station hygiene and to use fresh food from reputable sources. Also a good way when you have multiple feeders in your garden is to rotate feeder positions to avoid accumulation of waste or food droppings, and of course to provide water on a daily basis. This, of course, all sort of relies on us being watchful and trying to raise awareness about hygiene and precautions for the well-being of wildlife that visits our garden, but in the end also for the well-being of ourselves. Now, very briefly, some insights into our hedgehog health investigations. So the UK's only spiny mammal, and also the only mammal that our project deals with, it's regularly voted the most favorite wild mammal. However, despite its popularity, sadly, no matter which way we look at it, because as we can see in this graph, each of these lines represents a different survey using different techniques to estimate the national hedgehog population. British hedgehogs are in trouble, and they have been rapidly declining over the last 25 years. And it is now estimated that we have probably lost around a third, maybe even as much as half of our hedgehog population. And very recently, the hedgehogs have also been listed as a vulnerable species by the IUCN. So, to our current understanding, this is likely to be a multifactorial issue, most likely including things like habitat loss, habitat fragmentation, and of course also road traffic accidents. And we within the Garden Health Project, we're trying to look into whether any diseases might also be a contributing factor to this overall problem that the hedgehog population is facing. There is already a lot of knowledge on hedgehog health from individual animals, which are mostly presented as wildlife casualties to rehab centers or wildlife hospitals. But there remains an information gap regarding the diseases that affect hedgehogs in the wild, and whether they could be, whether these diseases could be contributing to the observed population decline in the country. So what we found so far is that whilst hedgehogs are infected with a variety of infectious diseases to various degrees, we very commonly find non-infectious conditions to be the ultimate cause of death, mostly due to mammal predation or road traffic accidents. However, there is still very limited information on this, but lots of discussion whether certain toxins might have also a negative impact on hedgehogs. And we have for many years now collected samples to further investigate whether, for example, rodenticides or slug pellets might be a cause for disease or even death in hedgehogs. And yeah, we're hoping to use those samples and test for that in the future. So what have we learned about hedgehogs so far? So we do So we have learned that there are non-infectious causes of death, that those are very common within hedgehogs, but also that they carry a variety of different infectious disease conditions. And we have to be aware that some of these might have a zoonotic potential, meaning that they can also affect humans or companion animals and livestock, which is why it is always important to follow good personal hygiene precautions, like wearing gloves when handling hedgehogs, or any equipment like food bowls that we might provide them with, and which might have been in indirect contact with the animal, and to always thoroughly wash our hands after we've handled any of these equipments for hedgehogs. We need to further investigate how our use of pesticides, rodenticides, or slug pellets might be affecting the overall health of the hedgehog population. And very importantly, to date, we have found no evidence of a single disease condition affecting our hedgehogs on a population level. To summarize, citizen science as a tool to monitor the health and diseases of garden wildlife. So we have learned that citizen science is a very powerful tool to conduct this kind of disease surveillance, and the diseases are very highly dynamic and often very unpredictable, so they require close monitoring over longer periods of time. And in order to differentiate between an endemic, meaning a well-established disease, or an emerging, so a new disease, we need to monitor over longer periods of time. And whilst we gain a lot of information from the opportunistic or ad hoc reports, in order to understand whether a condition might have an impact on the population of a certain species, we need to combine these reports with systematic data. And finally, sample archives are very crucial to facilitate the study of pathogens or diseases, to trace them in space and time. And I think the most important thing is that, especially when working with the public, collaboration and communication are the actual key to success for any such project. I want to quickly say thank you to my colleagues Becky Lawson and Joseph Heaver, and of course our partner organizations, BTO, the RSPB, Froglife, and SSL, and of course to our funders. And I wanted to leave you with some information on future events that might be of interest. One coming up in January, which will focus on pathological investigations, including a session also on how we conduct our examinations within the Garden Wildlife Health Project. And then there will be another great talk on citizen science again, and how it supports wildlife conservation, and that will happen in February. So more information on this can be found on the ZSL website, and lots of information on our project, the Garden Wildlife Health Project, can be found on our website. Yeah, and I hope I could give you some insights into how our project works and how we can learn a lot when we just work together and collaborate wildly. And yeah, fingers crossed I can answer any potential questions you might have. Thank you.